Hello, good afternoon, everybody. My name's Duncan Murdoch. I'm going to be chairing this session 13A live from Oxford, part of the Palaeontological Association virtual annual meeting. So we've got 5 fantastic speakers lined up for you. A little reminder to everybody, please put your questions in the chat as soon as they occur to you so that I can ask them to the speaker at the end of their presentations. And if you could click the little button that marks them as a question on the right hand side, then I'll know I can ask them to ask the speaker that question when they have finished. So we've got 5 15-minute talks, 12 minutes plus some time for questions. Any issues with reconnection, with the connection dropping out, there's a reconnect button at the top of your screen. Just give that a click and you should connect right back in. Okay, so we've got about a minute to go, so now's a good time to say hello in the chat, let everybody know that you're here. Yeah, good afternoon, Thomas. Thanks for jumping in. So, Nico, if you could turn on your camera and your microphone, I'll pull up your slides. Okay, so our first talk is Nico is going to be talking to us about morphological clocks and fossil tip dating. Take it away, Nico.
Hey, thank you so much. Thank you all for being here. So let's start. Fossils are the main source of information regarding the timing of evolutionary events, and as such, they play a crucial role in understanding evolutionary history, especially at deeper timescales. Now, technically, exploring evolutionary history can be done using data exclusively from living organisms, as well as some sort of understanding of their phylogenetic relationships. But it's been shown that the incorporation of fossils into trees of extant taxa is, of course, not only necessary for divergence time estimation, especially using more modern chip dating approaches, but it's also necessary to perform accurate macroevolutionary inference. And to paraphrase a recent paper by Lloyd and Slater, the results of this kind of analyses range from untrustworthy to nonsensicals if we do not account for fossil terminals. Now, it's evident that there's a lot to be gained by incorporating fossils into phylogenies of living organisms. But exactly the consequences, topologically speaking, of doing so are poorly understood. So this year actually, we published a paper where we show that fossils always favor topologies that are not found in the analysis of only extant taxa, so that the incorporation of fossils is actually significantly modifying the resulting topologies that we obtain from phylogenetic analysis of morphological characters. But given that this analysis was based on empirical matrices, we were unable to test whether topologies that include fossils were getting significantly better or significantly worse. Now, of course, fossils can only be included in phylogenies using morphological data, and as such, there's been an intense scrutiny in the last decade or so of the adequacy of alternative methods to reconstruct trees, especially focusing on comparing the consequences of choosing maximum parsimony versus Bayesian approaches. But a recent study has shown that this decision, whether to use Bayesian approaches or parsimony, the topological consequences of this are relatively minor. You can see in those bar plots, the orange represents the topological difference between the results obtained from either one of those methods, compared to the effect that it has if we include stratigraphy and perform tip-dated inference, which are the difference respect with the undated inference, either Bayesian or parsimony, are shown in sort of purple colors. So whether to tip date or not our morphological tree seems to be the most important decisions to be taken when performing a morphological phylogenetic analysis, and yet the consequences of this have been poorly explored. So what we're actually lacking, I would say, is some sort of understanding of how different inference methods sort of interact with different levels of fossil sampling and different amounts of missing data, and simultaneously these 3 factors determine our ability to accurately reconstruct phylogenetic relationships. And so this is exactly what we sought to answer in this study. and with it, hopefully, improve our understanding of these important questions in phylogenetics that people have been asking for decades. That is, whether fossils improve phylogenetic inferences of morphology, and whether stratigraphy contains useful phylogenetic signal. And so to do so, we turned to simulations. We simulated binary morphological characters using TrevoSim, which is part of the PaleoWare assembly of packages of software. And with it, we simultaneously simulated phylogenies and character data sets in the presence of natural selection. And one of the most interesting peculiarities about this program, and why we chose it over other available options to simulate morphological data, is that neither tree nor morphology are simulated under models that are later employed for phylogenetic inference. And although we know that Markov models are sort of very adequate approaches to model morphological characters, and we can also use birth-death processes to model the diversification history of organisms. But we also know that empirical data sets often, if not always, deviate from this simplistic models. And so the analysis of morphological data always includes some sort of element of model misspecification. And we sought to incorporate, similarly in this analysis, realistic levels of model misspecification as well. The data sets that we constructed contain 300 parsimony informative characters and 300 taxa, half of which were extant, half of which were fossil on average. And the trees, the parameters of the simulations were chosen so that the trees had a very specific shape. And as you can see this on the right, there's an example of one of the trees we used. These had always 5 major lineages, which connected deep down in the tree in a sequence of very short and deep internodes that's reminiscent of a tree shape known as an ancient rapid radiation. And the reason why we chose this type of tree shape is because it's been claimed in the literature that this might be a condition in which fossils could potentially help us a lot in reconstructing evolutionary relationships. But this is another piece of the puzzle that has never been actually tested. And so we simulated 250 of these data sets, and from them we subsampled down to 100 terminals, but we did so in such a way that we ended up with a sampling that contained exclusively living organisms, exclusively fossil lineages, or some condition in between. So we generated 5 levels of fossil sampling. We further added another level of complexity to this by incorporating 3 levels of missing data, including none, low, and high, which are the most realistic of all. And by that I mean that they've been sort of validated by a comparison with empirical morphological data sets. And we analyzed all of this data, which added up to more than 10,000 data sets, using 3 different methods of inference: maximum parsimony, undated Bayesian inference, and tip-dated Bayesian inference under the fossilized birth-death model, which I'm going to refer to as MP, BI, and clock, respectively. And so we obtained a series of inferred topologies, which we were able to compare back to our true simulated relationships, using metrics of both precision, accuracy, and also some sort of summary of the two, which are where we chose tree distances. And it's important to know that for precision and accuracy, we actually implemented both bipartition-based and quartet-based definitions of these. And the reason why we did this is because bipartitions are going to give us some sort of overall broad summary of what's going on across the entire tree, while quartets are mostly going to focus on what's going on deep in the tree. And this is evident here in this random tree that I'm showing you here on the right. You can see the deepest branches of this tree are colored in sort of dark red colors, and those deep branches have a high number of descendants in the x-axis, and therefore account for a large number of quartets on the y-axis. And so when we're using quartets, we're mostly zooming in and narrowing onto what's going on deep in the tree, while when we're using bipartitions, we're sort of getting back an overall summary of what's going on across the entire topology, regardless of its relative position. And a lot of the work that we did was actually ground truthing the simulations against 12 empirical data sets, making sure that they displayed properties that we thought were needed to be replicated for our conclusions to be applicable to the analysis of actual morphological data, including similar rates of evolution, distributions of branch lengths, levels of treeness, like I said, similar amounts of missing data, but also certain parameters that need to be fixed for the performance of tip dating inference, such as tip age uncertainty and tree height uncertainty. They were all obtained, mined from paleontological databases. And so onto the results. What I'm going to show you first is the results of accuracy. So when we're looking at this, we're sort of getting a sense of the proportion of correct statements that trees are making. And what I'm showing you on the left, we can see bipartitions on the top, quartets on the bottom. MP, BI, and clock are the three methods of inference. And the data that I'm showing you here in yellow is before we include any type of missing data. What you can see is that regardless of method of choice or the way we measure accuracy, maximum accuracy is always attained when we either sample 50% fossils or 100% fossils. So fossils seem to be improving accuracy relative to no sampling of fossils, so just considering extant terminals. When we incorporate the effect of missing data, which you see here in purple and pink, there's not much of an effect. What I've told you here before, 50 or 100% fossil sampling leading to maximum accuracy, is still true in the presence of even high levels of missing data. So a minor effect only of missing data on accuracy. For precision, and here we're dealing with sort of the number of statements, overall number of statements that a tree is making, or some sort of general level of resolution of these topologies. We see an interesting pattern in which we see on the top for bipartitions an increase in precision, and on the bottom a decrease in precision if we measure it using quartets. So this sort of opposite trends, what's telling us is that when we add fossils to trees, these fossils are sort of increasing the overall number of resolved nodes, the general level of resolution, but they're at the same time collapsing deeper relationships. And so we see this decrease in quartet-based precision. These deep relationships are resolved in their absence, in the absence of fossils, but tend to be collapsed when we include fossils. And once again, when we incorporate missing data, there's not much of an effect except under specific conditions. For example, the analysis of entirely extinct lineages and their maximum parsimony gets hit really strongly by the incorporation of missing data, but the general trends are still the same. So it's hard to get from this a sense of which model is actually outperforming the others. So to do this, we relied on quartet distances. So here we're comparing the true tree and the inferred tree using some sort of distance metric, which we chose quartet distances. And what I'm showing you in the three boxes are the three methods of inference across the 15 conditions that we explored, 5 proportions of fossil terminals on the x-axis, and 3 levels of missing data on the y-axis. And in white colors, I'm showing you short distances between inferred and true trees, that is good topologies. And in dark blue colors, large distances or bud results. And so you can clearly see that across all the conditions that we explored, probabilistic methods outperform parsimony. They find trees that are more similar to the inferred ones, to the true ones. Sorry. Tip dating in generally is significantly better than undated Basin approaches, especially specifically in all the conditions that are here marked with asterisks. So under all of those, incorporating stratigraphy and tip dating outperforms undated approaches. But maybe more importantly, we find that the best trees across all conditions are always found when we tip date matrices that contain a combination of both fossil and extant terminals. So neither entirely fossil sampling, not entirely extant sampling, but a combination of the two seems to be sort of the golden ticket. So it seems that fossils and extant terminals are sort of interacting positively with each other, and to further explore this, we asked whether fossils might be helping to resolve relationships among extant taxa and possibly even helping us resolve this set of ancient and rapid radiations that we incorporated. And for these, to test this, we subsampled our true and inferred trees down to just the extant taxa. And we subdivided all nodes in this topology into 3 bins of equal time length. And so we have a deep, a mid, and a shallow set of divergences. And for each of the nodes that fall into each one of these 3 categories, by comparison with the inferred topologies, we estimated the probability with which they're correctly resolved, incorrectly resolved, or just left unresolved, as you can see here on the right with this huge polytomy. And by repeating these across different levels of fossil sampling, we can sort of, and comparing how these probabilities change, we can get a sense of how fossil sampling is modifying our ability to correctly resolve relationships across extant organisms of different divergence times, and also specifically whether fossils are helping us resolve this set of deep and nodes associated with the ancient and rapid radiation. And so all of the data is here. There's a lot to take in. So I'm going to ask you to just going to zoom into this bit over here. What I'm showing you here is the proportion of correctly resolved nodes connecting extant taxa. As we sample more and more fossils, these box plots become sort of more purple. And what you can see is that fossils improve the accuracy, that is, they increase the proportion of correctly resolved nodes connecting extant taxa for both mid and shallow relationships. You can see here with these yellow arrows across all three methods of inference, the accuracy of these relationships improve. But there is actually no improvement in the resolution of deep relationships. So fossils don't seem to be helping us correctly resolve ancient and rapid radiations. What they are doing, however, is they're collapsing these deep uncertain relationships, which in their absence are generally incorrectly resolved. So here on the top, in the middle, actually, you see the proportion of incorrectly resolved deep nodes. And you can see this incorrect proportion decreases as we add more fossils, while the proportion of nodes that are just left unresolved in a polytomy increases. And this specific targeting and collapsing of deep uncertain relationships is something that we see exclusively under tip dating methods. There is some similar trend across Bayesian inference and maximum parsimony, but nothing to the extent that we see for tip dating. And this might be a big reason why this method is outperforming all of the rest. And so to conclude, we find that fossils improve the phylogenetic analysis of morphological data, even when they display high levels of missing data. So even highly fragmentary fossils seem to be improving phylogenetic inference. And they do so by improving the overall accuracy of the topologies that we infer, increasing the number of resolved nodes, but also by inducing the collapse of deep and uncertain relationships that are otherwise, in their absence, reconstructed incorrectly. We also find that tip dating under the fossilized birth-death process outperforms other methods of inference. Specifically, we find that the temporal information contained in fossils has useful phylogenetic signal, and that the best trees that we were able to recover are always found when all of the available information is combined, including the morphology of both extant and extinct taxa, as well as their stratigraphic information. And with that, I would like to thank my awesome collaborators. We recently put online a preprint of this that I'm going to share a link shortly. And thank you all for being here.
Thank you very much, Nico. I'm afraid there's no time to answer the questions, but there are some of them waiting for you in the chat. So if you'd like to head over there and answer them, if I can ask Lorenzo to turn on his microphone and camera, and I'll hand over to Lorenzo now. Take it away.
Thanks for the introduction. Thanks to watch my talk about the new Euchelicerate from the Fezouata Biota. So the Fezouata Biota is recovered in an area on north of Zagora. There are several outcrops spread around this area, and Zagora is on the southern east of Morocco. And all of these sites fall in the early Ordovician, in the Thermidorian. And as such, they are the Fezouata is a key site to study what occurred during two of the most important biological events that occurred during the Paleozoic. So the Cambrian explosion and the Ordovician radiation. The Fezouata shell formation is a basal shell type Lagestaten, and it provides us lots of fossils that preserved soft part as well as sclerotides part. And among the most common fauna, we have echinoids and graptolites, but also arthropods are abundant. As you can see in this slide, we have some chelionellids, we have some marellomorphs, canids arthropods, xiphosurans, and aglaspids. But this talk is about an Euchelicerate, so let's have a brief look to what the Euchelicerata clade is. They are a successful group of arthropods. They are characterized by the presence of a first pair of specialized appendages, the chelicera, and from the progressively fusion of the body into different agmata, the prosoma and the opistosoma. Having a look to them phylogeny, on the left tree you have a more broad-scale phylogeny, which include also their arthropod taxa, and you can see in this phylogeny that they recovered Abelia and Santacaris from Cambrian as sister taxa for the Euchelicerate. Instead, on the right phylogeny, you can see the division of the Euchelicerate in Prosomapoda, which are basically including all the Euchelicerate except of two synziphosurine, Ophacolus and Ibasterium. So I would like you to focus on Ophacolus and Ibasterium as well as on Abelia, because they are going to be part of our discussion. The reason why one of the reasons why Ibasterium and Ophacolus are not considered to be Prosomopoda is because Prosomopoda share uniramus appendages in the prosoma, and this is not the case for Ibasterium and Ophacolus, which show biramus appendages in the prosoma, as you can see pointed out by the arrow, in green the exopod and in blue the endopods. Even if there is still some debate about the true nature of these biramus appendages, because looks like they are they do not share the same insertion in the body, so they could derive from different anatomical parts. But the Ibasterium and Ophacolus are not the only arthropods that show this feature. As you can see here, we have Abelia, and also Abelia has biramus appendages under the prosoma, even with a slightly different morphology. And the exopod are considered to be stenopodus, basically elongated, like the one in the Ibasterium and Ophacolus. Another interesting things to note in Abelia is the opistosomal appendages, which are divided in two main parts, the distal part and the proximal part. But let's have finally a look to our new taxa. As you can see here from the general dorsal anatomy, the body is divided in a prosoma and in an opistosoma. The opistosoma is made of 11 targite. The first one of them is fully developed, which is something not usual in synziphosurine, that usually show a reduced force targite. Then the opistosoma is divided in a preabdomen and in an abdomen, and there is a telson spine, in the end. Other interesting features that we can see in this picture are the prosomal rim, a bipartite targite, the subaxial node, and the pleura. But especially the bipartite targite is interesting because it really resembles the pattern that we can see in Abelia, with a more sclerotized part of the targite and a less sclerotized part of the targite, well visible, especially in lateral view. But we say that the appendages are really important for the classification of those animals. So let's have a look to the appendages. This is one of the first specimens that give us insight about the anatomy. It's not possible to see each single appendages, but it's possible to recognize the number of them and how they are allocated in the body. And we have 6 appendages in the prosoma, and the 7 appendages belong to the first targite of the opistosoma. To get more information about the appendages, we decide to do a synchrotron experiment on the specimen that you can see on the top left corner. We chose this specimen because being almost completely enclosed in the matrix was a good candidate for the synchrotron experiment. And those are some of the results we get. On the left, there is a slice of the CT scan we did, and on the right, there is a 3D reconstruction, even if the specimens are quite flat and there is not a lot of depth in the 3D reconstruction. But you can see in lighter color the inner rami, and in the darker color the external rami. So this was the first in zinc that the appendages are biramus in this taxa. Then we were able to study this other specimen, and as you can see in this drawing, we have the exopod and the endopods, as well as the chelicera, and it's visible also the seventh pair of appendages, which actually belong to the opistosoma, and it's similar in shape to what we have in Ophaculus and Dibasterium. Another interesting feature about the appendages anatomy came from this specimen, in which we were able to see what looks like to be a common basipod for the two rami. And this, as we said, it's something different from Dibasterium and Ophaculus. In the end, this other specimen give us the chance to study the anatomy of a single exopod, giving us the chance to compare it with the exopod of Ophaculus. And if you have a look to them, you can see that they are quite similar in the anatomy, with a double spine on the preterminal podites and a seta on the terminal one. Then, speaking about the opistosomal appendages, we were able to understand that they looks to be more elongated than what we can find in the Dibasterium, preserving a distal and a prosomal part. Here you can notice, you can see the comparison with the Dibasterium and the distal and proximal part we have in this specimen, which is situation which we can most likely find in Abelia, which show a comparable anatomy of the opistosomal exopod, even if we do not have any trace of endopods in our new taxa. Then the last ventral feature we were able to study is what we right now think is an anal pouch. Our first guess was to this feature to be related with the aglaspid postventral plates, but after some discussion with colleagues and after reflecting that postventral plates are sclerotides feature that should preserve more often, when instead we had few specimens showing this character, we start to think that was most likely a membranaceous structure like the anal pouch in Abelia, and also considering the other affinities between those two animals, we decide to interpret this in this way. In this slide, you can see a comparative reconstruction of new taxon, Dibasterium and Abelia, with color used to show the potential homology. And the reconstruction we did of this taxa came from not only the specimen that I show you right now, but we had the chance to examine more than 300 specimens, being this new taxon one of the most common in the Fezouata. So to test if there were some affinity with Abelia and as well as with Ophaculus and Dibasterium, we at first decided to run an analysis using the matrix from Parry et al. 2019, which is a broad matrix. So we were searching for a broad confirmation of this new taxon to belong to the Euchelicerate. As you can see here, we get this monophyletic group with Ophaculus and Dibasterium, and that's what we were pretty much expecting. But then to test in a better way the relationship with the other synziphosurine and in general with the Euchelicerate, we decided to run another analysis, starting from Lambsdale et al. 2013 matrix and implementing it with Abelia and coding Dibasterium from Seldon et al. 2015 and having some review to Weinberginia, which was an important synziphosurine as well. And as you can see here, we end up with again a monophyletic group with our new taxon as most basal of this nucleate, and Abelia outside all the proper Euchelicerate. So to conclude, we are showing you this new taxa, which resolve as a monophyletic group with the Bacterium endophagorus, accumulate by the presence of Biramus appendages with Stenopodus exopod. But on the other side, we found also some similarity with the Cambrian arthropod Abelia, such as the anal pouch, the morphology of the opistrosomal exopod, and the bipartition of the targite. I would like to thank you, all the people that collaborate to this work. Of course, all the people that give us advice and help us in different way, as well as my lab, which is a really good thing to be part of. Thanks.
Brilliant. Thank you very much, Lorenzo. So we have some time for a quick question. There's one in the chat from Mario. So are there supposed functions for anal pouches?
So it could be something related with the reproduction, I guess, being the position, or it could be something related with the waste accumulation, which is something that is present around some Euchelicerate. But yeah, we didn't think too much about the possible function of the anal pouch.
No problem. Thanks, Lorenzo. It's a question from Russell Garwood. It says, lovely stuff. Sorry if I missed it, but do you have any feeling about the number of segments in the chelicerae of this new fossil?
So no. So far, no. All the time we found the chelicerates. I don't know if you had a look to the scale, but they are really, really, really small animals. It was almost impossible to distinguish between the number of segments in the chelicerate.
Thank you. And I'll take one more from Rudy Lerisier-Brill. He says, did you try to use the matrix of the Aria and Quran 2019? That's the Mollisonia paper.
Actually, is one of the matrix that we used. We didn't try to remove Mollisonia. If is this the question? Matrix? Yeah, it's the one we used. It's the one in the previous one I showed. Duncan, we cannot hear you.
Thank you. Any more questions? I'm sure we'll get them in the chat. So thanks a lot, Lorenzo. So if I can ask Christopher to join us. Okay, so our next speaker is Christopher Whalen, searching for macroevolutionary early bursts. So take it away.
All right, thank you. And thank you all for signing into this session, and for the event organizers for putting together, I think, a pretty well-run conference, all things considered. So I'm going to be talking over the next several minutes about some work that Derek Chelly and I recently published in Science Advances, testing for macroevolutionary early bursts through the lens of the Paleozoic ammonoid record. So, I'm sure I don't need to explain the early burst model to anyone here. It's fairly well known. It's one of these concepts that everyone learns in introductory evolutionary biology and paleontology. As you can see here on the left, some classic work by Foote looking at Paleozoic crinoids. It's this idea that early on in a clade's history, or at the onset of an evolutionary radiation, while diversity is still low, disparity can be disproportionately high. As you can see, it doesn't necessarily need to be higher than later on in the history of the group, but it does need to be higher than expected given diversity. And you can establish that expected ratio from the figure on the left just by looking at how diversity increases and disparity stays relatively flat for crinoids. On the right, you can see, cichlids, a classic recent example. And in the figure on the far right, you can see a scenario where disparity is actually decreasing through time. And that decreasing through time is often overly conflated with the early burst model. And we talk about bottom-heavy clades, like you can see here in this Hughes et al. Study. But I do want to make the point that early bursts do not require that a clade be bottom-heavy. The requirement is that disparity is exceptionally high given the lower diversity at the onset of a radiation. that can manifest itself as a bottom-heavy clade, like you can see here, but it doesn't need to. And this is, of course, a very important question because some, such as this youth study, have posited that it may be one of the very few macroevolutionary rules that we have. That all said, there have been a number of challenges to the early burst model lately in the literature, especially using phylogenetic comparative methods, such as this Hopkins and Smith study on the left looking at echinoids. They found that when you look at the group overall, you do not see an early burst, although they did find that you may see them within specific subclades that feature a unique adaptation that allows them to radiate. And this is an idea that's carried over by Slater and Fisher in this 2019 study they did looking at mammals and similarly looking for evidence of early bursts. And they argue, you can see here on the bottom, the actual symbols don't matter, but you can see here on the bottom different traits that they're measuring through mammals. And they're looking at whether each individual trait is showing you evidence of an early burst. The point is that the only traits that are going to show you an early burst are the ones that are relevant to the ecology of the animal, relevant to an early burst. And so they argue that perhaps these phylogenetic comparative methods are not finding evidence of early bursts because they're looking at the wrong traits, or perhaps they're looking at the wrong scale of evolution. They're looking at too small and too young of a clade. And so this is what we sought out to test with ammonoids. Ammonoids, of course, have a very well understood, well resolved stratigraphically fossil record, but also very importantly, by looking at the coiled fragment cone, a planisspiral fragment cone, you can very simply get disparity measurements without having to deal with landmarks and semi-landmarks, etc. And from these shell shapes, you can directly predict the paleoecology, and this is the important part. Because the coiling of the shell imposes fundamental hydrodynamic properties to the animal, there is a paleoecological, a direct paleoecological connection here that we can establish between the shell coiling measurements we make and paleoecological niches that these cephalopods could fill. Now, this was theoretically shown in this past image here from some work that Westerman did in the 90s, but it was mathematized by Ritterbusch and Botcher in 2012. Here, what they've really done is combine the sort of Raupian coiling parameter type measurements in such a way that you can actually place a coiled fragment cone in Westerman morphospace, this ternary diagram you can see on the right. So it's a mathematized version of the figure that you see on the left here. We have a quantitative ecomorphospace. So what we did was measure 749 Paleozoic ammonoid species. These span about 156 million years of ammonoid evolution from their origination in the Eocene to the end of the Induan. The Induan, so that we include the recovery from the Permo-Triassic mass extinction. You can see here it's color-coded based on the different ammonoid orders. That's just a visual thing. It's not particularly important for what we're going to be talking about because these orders are not monophyletic. They are for the most part, paraphyletic, and so they aren't very useful for asking the questions of early burst dynamics. We really need to look at ammonoids as a whole, so we ensure we have a monophyletic clade. And all of the actual primary measurements for this are in the supplement to this paper, and I encourage you, if you're interested, to look that up. Now, at the simplest level, we can take those zones in Westerman morphospace and plot the relative proportion of different paleoecologies in ammonoids over time. And you can do something like this, and you see here on the bottom in yellow that in the Devonian, nectic ammonoids seem to have been more common than later in the Paleozoic. And when we get to the Fumenian, planktic ammonoids become much more prevalent. And that's simply based on the lines that are drawn by Ritterbusch and Botcher in this figure on the left. That's really what the big uncertainty is in Westerman morphospace. There is empirical simulation evidence that you can see here on the right, that you can't get around the fact that these shells impose hydrodynamic properties to the animal. And so there is an aspect of this that, say, an organism with a high TH value, a spheroconic shell in the far right, is going to have a lot of drag and be a rather inefficient swimmer, and so must consequently be planktic. You can see that the lines don't correspond one-to-one to the Western morphospace on the left, but that's not really relevant to our discussion of early bursts. What is relevant is that this clearly is a paleoecological signal. Now, I basically measured what I could get my hands on from museum collections and from the literature. But it does turn out that this, and this is important, this is a representative sample. So here you can see in black the measured richness through time, species richness through time, resolved to 1 million year stratigraphic bins. And on the right, you can see the in blue, the comparison to the Ammon online database. This is maintained by Dieter and colleagues. It's a very well maintained, well curated database of Paleozoic ammonoids. Now, there's a bit of a hiccup in trying to make paleoecological measurements from Westerman morphospace, and that is that we're dealing with compositional data. So we've taken these primary shell measurements, we've then calculated our coiling parameters, we've then normalized those relative to one another to put them into Westerman morphospace, and we then need to transform that back into Cartesian space if we're going to make any measurements. And this is simply because in an HSN simplex, such as Westerman morphospace, moving from, say, 90% to 100% is not the same as moving from, say, 10% to 20% in one of these measurements. So you do have to make that transformation. This is rather inconvenient and somewhat removes your results from your original measurements. It would be much simpler if we could look at principal components directly derived from those primary shell measurements. The problem with this is the principal components aren't necessarily ecologically relevant to the same extent that Western morphospace is. But as it turns out, PC2 does correlate pretty well with the U parameter and PC3 with the TH parameter. You can see here PC1 doesn't correlate particularly well with any of them, and we don't have a great signal for W in PC space, or at least not in the first 3 principal components. So we can use these 2 principal components as our proxy for paleoecology. So then the question is, remember, we need disparity that's exceptionally high given diversity. And you don't want to do just a simple one-to-one comparison. You don't want to divide disparity by diversity to try to get that at that measurement, because if you picture the PC space or the Western morphospace, the first few points that you add when there is nothing there, the first few species you introduce into an ecosystem have a lot of room to differentiate from one another, to create a larger and larger convex hull. The larger that convex hull is, the more species you have, the harder it is for each new species to differentiate from the other ones, right? So it's not a linear curve. There's an asymptote there. So we built a pretty novel, I think, null model to try to address this. We take our species list and the stratigraphic ranges associated with those species, fix that, and then randomize with replacement the PC measurements, PC2 and PC3, from the actual data set. Do that 10,000 times, take the median, and we have our null expectation. So here's the results of that. Above you can see the actual result, and below you can see the residual taken from comparing the measured disparity through time, the black line above, to the null median expectation, the white line above. In dark blue, we have the 50% envelope. This is from looking at the first and third quartiles. And then in light blue, the 90% envelope, looking at the first and 19th ventiles. The strictest sense, you could say, for an early burst, we want to see during irradiation disparity exceed the 90% or residual disparity exceed the 90% confidence envelope here, or I shouldn't say confidence, the 90% envelope here. And at a simple glance, you can see that that never happens in any point along this curve. Very rarely, in fact, does disparity exceed the 50% null envelope. So even if you want to take a somewhat more generous, less strict interpretation for what we should see in an early burst, you're still not seeing them. There's three intervals where disparity is above that 50% interval. The first one in the early Jurassic, that's just a pure chance anomaly because there's 3 ammonoids with very extreme morphologies that happen to overlap for that 1 million year. It's the last appearance of 2 of them and the first appearance of 1 of them. So that only occurs right there. You can see that our other high disparity is in the mid-Toarcian and late Toarcian and in the mid-Fezouata, notably not following the mass extinction. The mass extinctions here are in red. They're wide bars because this is taken from the Gradstein et al. Older stratigraphic scale, and the mass extinction intervals aren't resolved quite as. Now, just to briefly walk you through this, at origination, you can see that disparity is well within the null 50% envelope. It does not exceed it. We do not have an early burst in the strictest sense of the initial radiation of ammonoids. I also think, though, that recovery from a mass extinction, especially for ammonoids, is another scenario where you expect an early burst. It's sort of a proxy or a replication event of these radiations, because ammonoids famously have this sort of boom and bust evolutionary dynamic where they're hit extremely hard in a mass extinction with 1 or 2 surviving species and then radiate quickly from that surviving stock. Now, you can see at the Angevetian extinction, similarly, we're near the null envelope. We do not exceed it in that post-extinction radiation. At the Endfranian extinction, this is the late Devonian mass extinction, one of the big 5. We see that in the recovery, despite richness recovering quite quickly, as you can see here on the right, disparity does not. We are well below the 90% confidence interval. So we are seeing the exact opposite of an early burst. We are seeing disparity exceptionally constrained, not exceptionally high. This is surprising. You might expect it to be within the 50% envelope, but well below the 90% is not something we expected. Looking at the Endfemenian mass extinction, you again see you're within the 50% confidence envelope. Looking at the mid-Serpicovian extinction recovery, you are just barely within the null 50% envelope there. The extinction itself brought ammonoids below the null 50%. Looking at the end Guadalupian mass extinction, you see a drop from the null 50% to below it within the null 90% in that recovery. And finally, in the end Permian mass extinction, it's a little hard to see here, but if you squint or move closer to your screen, you can see that in the recovery in the Induan, disparity is again below the null 90% envelope. So in our 2 big 5 mass extinctions, our 2 biggest radiations, in addition to origination, we are not seeing early bursts. And in those 2 recoveries, we are seeing disparity well below the expectation given diversity, below that 90% envelope. In other words, we are not seeing any evidence for an early burst, despite several different radiations where you could see it. And with that, I'd like to acknowledge some colleagues, and I'm sure I'm really pushing the time. So thank you.
Yeah, thank you very much, Chris. There's some questions for you in the chat which you can address during the rest of the talks. I'm afraid we don't have time for them now. So if I can invite Connor to join us. I'll bring up your slides. So now we have Connor O'Keefe, Palaeoenvironmental Reconstructions in the Lower Jurassic. Take it away, Connor.
Okay. Well, good afternoon, everyone. I'm Connor. I'm a third-year PhD student at the University of Leeds, supervisors Chris Little. And today I'm going to be talking about the research I have carried out so far in my PhD, which is on the Lower Sulphur. So first of all, some societal relevance for my project. that anthropogenic coastal dysoxia is on the rise in modern marine ecosystems. Dysoxia is where the dissolved oxygen content of water drops below 2 milliliters per liter. Since 1950, over 500 cases of coastal dysoxia have been recorded, and the rate is increasing. Although we can closely observe the response of spatio-temporally limited marine communities to dysoxia, the effect of projected widespread dysoxia on large-scale marine communities remains uncertain, particularly since going forward OMZs are likely to expand as well. So to better inform our predictions about the consequences of that, we need to look to the geological record, which contains multiple periods of widespread oxygen depletion, for instance, the oceanic anoxic events. I'm going to be focusing on the Toarcian oceanic anoxic event, which occurred 183 million years ago in the early Toarcian of the Lower Jurassic. This is characterized by widespread oxygen depletion in the European epicontinental seaway, partly as a consequence of the paleobathymetry, which was rather rough with loads of isolated subbasins, which had limited oxygen resupply and were very prone to the development of anoxia, especially if there is a concomitant perturbation to the exogenic carbon cycle, like there was at the TOE as a consequence of the Kourou-Ferrar large igneous province. Many of these subbasins became anoxic and developed vast accumulations of anoxic black shales. One famous locality is the Cleveland Basin in Yorkshire, UK, which is the focus of my study. And my samples in particular come from a locality called Horsker Bottoms, which is just north of Robin Hood's Bay. So the Cleveland Basin, I think I'll just walk through the stratigraphy quickly, just so you have an idea what we're dealing with. It's a succession of shallow marine mud rocks, which are divided into the grey shales, and overlying that, the Mulgrave shale member, which is deposited in the Euchelicerate subzone. I'll use that term for a talk, so it's useful you know what it means. The Euchelicerate subzone is also characterized by a negative carbon isotope excursion in marine organic matter, which indicates carbon burial, and also high levels of isoneritane, which is a carotenoid biomarker. It results from the breakdown of the cell walls of sulfide-forming bacteria, which require light, their photic zone. So the presence of this implies that you have hydrogen sulfide, euxinia, in the photic zone. However, in the Euchelicerate subzone, you see every so often these thin accumulations of low diversity, high abundance bivalve faunas, specifically the Pseudomytoloides dubius within the Euchelicerate subzone. In the upper grey shales, you see Zitra radiata as well. And although it's never been tested by high resolution geochemical studies, which is what I'll be doing, they've long been suspected to represent periods where these apparently persistent euxinic conditions were interrupted for a brief time, so for a period of years to decades, so that this bivalve community can become briefly established. In addition, in the underlying grey shales formation, you have these 3 decimeter thick black shale units called the sulphur bands in the mining vernacular. I'm going to be focusing on the lowest of these, the lower sulphur band. Now, at first glance, these appear to be homogeneous black shale units, but if you look at sub-centimeter scale, you see there's actually quite a lot of variability within them. This is the lower sulphur band. You see that the lower 5 centimeters has these hummocky cross-stratification features, indicative of bottom water current activity within a black shale, which is not what you usually expect. 5 centimeters of bituminous mud there, and at the top, it's thoroughly penetrated by chondrites and rhizocorallian traces. So part of the reason that you think these imply persistently reducing conditions is because previous studies on the sulphur bands and the formation in general focus on meter scale variability and not centimeter scale variability, which is where my work comes in. I'm looking at I'm taking a multi-proxy approach to studying both the shale beds and the sulphur bands at sub-centimeter scale, comparing ichnofacies and classic descriptive sedimentology with geochemical proxies such as iron speciation, a total digest, and some parambolic work as well. Won't have time to get into the Bocqueval. So here are some of the most pertinent results of my research so far. First of all, you see on the far left there, you have a photomosaic of the lower sulphur band. And as you can see, where you have this intensely bioturbated fabric give way to hummocky cross-stratification, bioturbation intensity abruptly drops, and remains rather low right up into that bituminous horizon where you have high levels of total organic carbon and sulfur as well. But interestingly enough, the recovery of the biota starts while conditions are still reducing, as indicated by the total organic carbon and sulfur plots. And what we think is one possible reason for this is that these chondrites traces actually represent later generations of bioturbators, which are percolating down into sulfidic sediments that have already been deposited. And the reason they're doing this is because they're sulfide farmers, farming the sulfide for chemosymbionts to generate energy that way. Some modern chondrites trace makers, like diacyride bivalves, make use of this. I haven't found any thiocyrids in my section, and this is speculation, but it just goes to show there's a plausible way of reconciling this. I also show pyrite rhamboids. Just briefly, for those of you who might not be quite so familiar, pyrite rhamboids are these spherical accumulations of pyrites that form. If they form in the water column, i.e. if there's water column Euxinia, they're consistently small because they can only grow to a certain size before they fall out. If they grow in the sediment, they can reach wider sizes, and their distribution of sizes is wider as well. And you can see that in the box and whisker plots there. I've marked the ones which indicate that they were deposited under euxinic conditions in black there. And just to show that better, here's a Wilkins plot which compares standard deviation with the mean. And you can see mostly in the oxic realm, but a few from that bituminous horizon within the euxinic realm. So probably there was Euxinia within the middle part of the lower sulfur band, although not much to go into the photic zone, as I'll show in a minute. Okay, now this is interesting. I also looked at the reduced iron proxy. Now, this is a very good redox proxy, but it's often misapplied. Taken in isolation here, it implies that conditions were persistently anoxic ferruginous. That is, conditions were persistently anoxic to the point where highly reactive iron was becoming persistently enriched. Now, that doesn't fit with the bioturbation data. And what we think, and some of the other geochemical data, which I'll show in a minute, and what we think is going on here is that the highly reactive iron baseline, that is how much highly reactive iron there is normally, regardless of oxygen state, was elevated within this sedimentary environment, probably because A, it was highly restricted, and B, because the surrounding hinterlands developed lateritic profiles. This was the lower Jurassic, after all, which was mostly a rather hot, humid environment. So it makes sense that there were lateritic profiles around the basin, weathering in loads of highly reactive iron into the system. Here's some other redox data. I've collected a whole lot more than this, but I don't have time to show it. But here are the most important ones. Sedimentation rate is rather constant, as the aluminium profile shows. You have manganese enrichment. That's strange for a black shale, because manganese, obviously, in anoxic conditions, is reduced to the 2+ state, leaves the sediment, and normally leaves, so you have manganese depletion. But if you have a highly restricted environment, it's got nowhere to go. It just accumulates there until it can bond with carbonate and become removed and enriched with the sediment. So that's what I think is going on here, high restriction under dysoxic conditions. And you have molybdenum enrichment as well, although not stuck to crazy levels. So I'm going to suggest that the geochemical switch and its bonding with thymolybdate doesn't actually occur here. So you have some possibly limited euxinia within the central interval here, but not quite enough for the quantitative drawdown. And here is where sort of the paleontological relevance comes in. I found, studying this section, an isolated individual of Pseudomycoloides dubius. Here it is, it's about 0.5 centimeter wide. And this is not actually an uncommon occurrence in the lower sulfur band. You often find Pseudomycoloides dubius here as individuals, never as those big accumulations like in the Euchelicerate subzone, but nonetheless you find them. Here's some concomitant redox data. The picture isn't entirely consistent because of all the different caveats with the proxies, but on the balance of probability, I'm going to suggest that individuals of Pseudomycoloides dubius could tolerate severe to extreme dysoxic conditions with oxygen concentrations between 1 and 0.2 milliliters per liter. There are modern bivalves that can tolerate oxygen levels this low, and this just goes to show that modern marine communities subjected to widespread to extreme dysoxia going into the future, we can expect them to develop their ecosystems to become low diversity, high abundance of these epifaunal organisms able to tolerate oxygen levels this low. So just to briefly go into some feature work and planning, I will apply these same proxies to the monospecific high abundance event beds and test whether the conditions concomitant with these communities are different than those with the single individuals I found in the lower sulfur band. So what factors are necessary for the formation of these high abundance communities? A recent paper by Pierre et al. In 2020 has suggested that these, they looked at halobids, which are have a similar mode of life, but they suggested that these sorts of organisms have planktonic larvae. And so what might be going on here is that in these intervals, opposite conditions are sufficiently present in the upper water column that you get high volumes of larvae reaching into the environment and colonizing the sediment surface. There's also this idea of a sediment stabilization positive feedback. If you get some of these thin bivalves forming on the sediment surface, they can start to grow and harden the sediment surface, which allows more to become established, and you get positive feedback of sediment stabilization, allowing this pavement, for lack of a better term, to become established. This might be assisted if the idea of Caswell and Co. is holds water, that these bivalves are also nutrient limited. And if you have enrichment of various trace elements, for instance, molybdenum, that can actually facilitate these organisms in growing larger shells. And you can see how that accelerates the positive feedback even further. So these are some of the hypotheses I intend to test in this future batch of my PhD work. But to summarize what I've talked about here, the lower sulfur band is lithologically heterogeneous, and this reflects rapid changes in palaeoenvironmental conditions, which is weird for a black shale deposit. A multi-proxy approach is key to understanding what's going on here. And crucially, a multi-proxy approach is required if you're using the iron redox proxy, because it's very good, but it cannot be uncritically applied. My deposit was you need an understanding of the basin hydrography and factors such as how much reactive iron was in the system. In order to make proper sense of the data, you need some paleontological data and some other redox data as well. I was able to eliminate that based on the multi-proxy approach. Pseudomytoloides dubius was able to tolerate severe to extreme dysoxia, and a better understanding of the response of these communities in the past will better prepare us for the consequences of marine oxygen depletion, widespread marine oxygen depletion into the future. Thank you very much.
Thank you very much, Connor. We have a couple of questions for you in the chat. The first one comes from Liam Herringshaw. This one will test if I've got my teeth in. Let's see if I can do it. So he says, great talk, Connor. Does the Rhizocorallium chondrites ichnofabric development fit with the Savdra and Boccia 1989 model of black shale bioturbation?
I should be more familiar with that, but I believe it does. It's one of the, I'm more familiar with Wignall's oxygen-restricted biofasces, and it's their biofasces 1 or 2. So a few benthic fauna disturbing the sediment surface, but not these high diversity communities. So my answer, possibly yes.
Okay, thank you. And we have a question from Rudy. Could the single individuals of P. dubius have been transported?
Probably not. We know that, the paleobiology of these organisms has been the subject of a wide variety of debate, but I think on the balance of evidence, they could have been transported on like driftwood that's floating in, but we do see vast accumulations of them where there's no evidence for large amounts of driftwoods coming into the basin. So it's most likely that they could have also just fallen onto the sediment surface and developed in situ. There certainly isn't, it's not required that they be transported. And we know in many cases where we do have lots of Pseudomytoloides dubius that they in fact weren't.
Brilliant, thank you very much. I think there's some more discussion going on in the chat, so I'll let you head over there and I'll invite our final speaker for this session, Will Crab. Get your slides up for you, Will. So Will is going to tell us all about his fantastic paraconodonts. So take it away, Will.
All right, thanks Duncan, and thanks to all the other speakers for an excellent session. I'm Will, I'm a PhD student at Leicester, and today I'm going to be talking about some exceptionally preserved Cambrian paraconodont elements and what these can tell us about the biology and diversity in the earliest vertebrates. So conodonts are an extinct group of eel-like jawless vertebrates that are known almost exclusively from the phosphatic tooth-like elements. They've been used extensively in the past for biostratigraphy and paleoclimate studies, and over the last few decades, they've emerged as quite a useful kind of tool to help study early vertebrate evolution. So the group that I focus on in particular are known as paraconodonts, and these are disarticulated phosphatic sclerites that are found from upper middle Cambrian to lower Ordovician strata, and they cover a whole wide range of forms and shapes, as you can see from the SEMs at the bottom of the slide. They're thought to be directly ancestral to euconodonts, or conodonts as most people know them, based on the structural homology between coniform paraconodont elements and euconodont basal bodies, with euconodonts differing with the addition of a phosphatic crown tissue, which I've illustrated with some real samples at the bottom on the left there. So as a result of this, paraconodonts are thought to be among the earliest vertebrate tissues in the fossil record, and can therefore act as vital tools to help us study diversity and biology of the earliest vertebrates. However, paraconodonts are quite poorly understood from a paleobiological perspective. That's because, unlike euconodonts, no body fossils of paraconodonts have been uncovered, and any natural assemblages of elements are limited to fused pairs of coniform elements, which is in stark contrast to natural assemblages we see in euconodonts. For example, the one on the right, where we see basically an articulated apparatus. So as a result, the apparatus structure of paraconodonts is completely unknown, and this directly hampers our understanding of the paleobiology of these animals. But nevertheless, studies of large populations of elements can help to yield patterns of biological variation. So, for example, Shaniaski noted ontogenetic sequences within two separate species of Furnishiner, which seem to develop from these small, narrow structures into these larger structures with flared bases. And more recently, Bagnoli and Stalger described intraspecific variants of Furnishiner holmi with differences in the element symmetry and curvature thought to reflect a separate position within an apparatus. However, studies like these haven't really been extended to any other paraconodont taxa, so paraconodonts are still predominantly classified by a form taxonomy. So the Deadwood Formation is a large succession of mudstones and sandstones found in subsurface Saskatchewan that covers from the middle Cambrian to the lower Ordovician. Drill core was collected through the Ferongian interval of the formation from the University of Regina drill core repository, and areas of dark mudstone, as can be seen at the top of the slide, were processed using a bespoke hydrofluoric acid treatment that allows for the extraction of larger and more delicate fossils than would normally be acquired through standard palaeontological processing. And from these drill core samples, we get this beautiful assemblage of paraconodont elements that are preserved as small carbonaceous fossils, or SCFs, where they've apparently become demineralized and flattened during taphonomy. This preservation leads to an exceptional visibility of their internal growth structure, which can be easily viewed under transmitted light. So this provides us with a huge data set and an absolutely unparalleled opportunity to study ontogeny within paraconodonts and to identify early and late growth stages to illustrate how divergent form taxa can often share direct biological relationships. So the aims of the study can be summed up as to compare this new assemblage to previously described assemblages in the literature and taxonomically classify it, and then developing from that, identify any potential ontogenetic sequences or stages within single species, and then ultimately discuss the implications of this for the paleobiology of paraconodonts. So the Deadwood assemblage contains over 1,800 individual conodont elements. A large component of these are this diverse coniform fauna. unlike other elements, these ones are more challenging to taxonomically classify due to their flattened nature, which precludes them from a three-dimensional analysis. However, there are some very distinct taxa, such as Mullerodus in the top left and Furnishiner by my webcam, which have a very distinctive outline morphology that allows us to classify them. And as well as paraconodonts, we see other taxa, such as protoconodonts in the top right, which are thought to be mouth parts of chaetognaths, and also evidence for euconodonts reflected by elements of proconodontus in the bottom left. A small component are these very distinctive laterally extensive forms represented mostly by Ceratocambria and Proacodus. Ceratocambria in particular is an exceptionally rare genus, which is described here for the first time outside of Sweden and China. And we also see some evidence for potential ontogenetic sequences within these elements, and I'll touch back upon that later in the talk. And well, probably the most characteristic part of the assemblage are these W-shaped elements of Westergaardedina. So we describe 5 separate morphospecies here, 3 of which are referable to previously described specimens in the literature. So Concamerata and Cavata on the left there have both been described here for the first time outside the alum shale in Sweden, and Tricuspidata at the bottom has been described in multiple assemblages around the world previously. And we also seem to have evidence for 2 potentially new morphospecies that I'll refer to here as species A and species B. So if we want to identify ontogeny within these elements, first we need a bit of a kind of appreciation of how paraconodonts grow and develop. So using this figure from Murdoch et al. As an example, we can see the growth and ontogeny and development of an element of Furnishiner using synchrotron data. So if you can see from D, the element starts as this small conical proto-element, and then gradually develops into H, a kind of adult functional form, through the addition of tissue to its base and sides in what we call basal accretion. So we can take this and apply it to elements we see in the Deadwood Formation. So for example, here, elements compared with Procodus obliquus and Mullerodus cambricus are seen to develop from these small initial elements into gradually larger elements through the addition of tissue to the base, and then ultimately forming the adult stages through the development extension of a lateral process and basal rim, respectively, showing quite a distinct allometry throughout their development. And then we can take this a step further in elements of Westergaardedina due to the exceptional preservation of growth structure. So we can use software such as Photoshop to kind of erase or crop out later growth stages in the element. The line marked there by the white dashed line. And then when we can do that to create a sort of artificial early growth stage within the element. And when we do that, when we get the artificial early growth stage, this can be directly compared with what would be an otherwise morphologically distinct element that's collected from other samples. So again, highlighting this really distinct allometric development within paraconodonts and a seemingly more complex allometry within Westergaardedina. And then again, also highlighting how the distinct form taxa can share direct biological relationships. But however, when we try and apply this method to other elements of Westergaardedina within the Deadwood, we run into a bit of a problem. So, for example, this element of curvata here on the left shows these weird kind of paradoxical truncating growth packages within its turning points, which, as you can see on the right, can't seem to be reconciled within a standard model of basal accretion. And indeed, when we look at typically preserved phosphatic elements, so here's one we collected from the alum shale in Sweden, that's being imaged through oil immersion techniques. we again see these truncating packages within the turning points, and again can't seem to develop these or reconcile them with a basal accretion model. So what are these truncating packages? Are they a product of growth or a structural part of the element, or are they perhaps maybe some sort of superficial feature as a result of taphonomic processes or surface remodeling? Well, we can test that using synchrotron-based microscopy. So we took a bunch of different elements of Westergaardina to the synchrotron at the Swiss light source to try and get a better understanding of the internal three-dimensional morphology. So in A, we can see an example of one of these paradoxical elements with these truncating growth packages. And in B, we've taken a section that bisects these lamellae. And then we can see that these lamellae kind of are pervasive throughout the entire element structure, indicating that they are, in fact, a primary growth feature and a structural part of the element, not as a result of taphonomy or later remodeling of the element. And in fact, we can then use that synchrotron data and segment out these structures using a mirror. So the structures there highlighted with these orange colors within the model, and they seem completely separate from the rest of the element, which is represented by these green kind of colored segments, which seem to develop through that kind of standard basal accretion that we've been discussing earlier. So this could potentially indicate that there's two separate stages or phases of growth within at least these particular elements of Westergaardina, which is something that's completely unseen within any other paraconodont elements, and could potentially highlight an entirely new method of morphogenesis within the earliest vertebrate skeletons. So this is ongoing work, something I'll be working on over the next year, and hopefully I'll have more results to present at a later date. So I guess just to conclude what I've been through today, paraconodonts can act as a vital tool to help us study and examine ontogeny and diversity within the earliest vertebrates. And morphologically distinct elements can be reconciled with the same species by examining ontogenetic development, which can help us to partly bridge that gap that exists between form taxonomy and a more biologically informative taxonomy. And finally, the paraconodont Westergaardina seems to show multiple methods of growth, which expands our understanding of the development of the earliest vertebrate skeletons. So I'd just like to thank the people listed below for their help with various aspects of data collection, and thank you very much for listening.
Thank you very much, Will, for a brilliant talk. We've got time for a couple of questions. The first comes from Paul Smith. He asks, does your data allow for any estimation of multi-element apparatus composition of paraconodonts?
Yeah, that's a good question. I don't know if our sample sizes are large enough to be able to do that. I mean, I've tried to kind of plot out and kind of see if there's elements that kind of co-occurring with each other within samples, or like regularly co-occurring, but I don't think I have enough like stratigraphic range or like number of individual samples to be able to reliably extract that sort of information.
And a follow-up question from Paul. Have you compared your SCFs with phosphatic deadwood collections?
Not properly kind of compared them, because I know Godfrey and Allen has extracted some from the deadwood formation floor, but in terms of the paraconodonts, they didn't seem to be particularly diverse from what I've seen. So this SCF assemblage seems to be a lot more diverse in comparison, but I've not really properly compared the two.
Probably maybe something to look at in the future. A question from Tom Hearing. It says, really nice array of images. Well, do you have a sense of how much of the Westergaardina W was exposed in life versus how much is embedded in soft tissues?
Yeah, that's an interesting question that I'm trying to figure out myself, because some elements, it seems pretty obvious because they have big, large basal cavities like we see in paraconodont elements, the standard coniform ones, where you'd expect it to just kind of be protruding out of the kind of epithelia. But then, so we can use cavities, I guess, to give an estimation of like where the element would have been extruded. But some seem to show really paradoxical cavities, like alongside the side of the element and then not on the bottom. So it's a really weird question, but it's one we're kind of trying to address as well throughout this project.
And finally, from Martin Smith, if Vesicardina grows differently from other vertebrates, then can you be certain that it is indeed a vertebrate?
Yeah, and that's another good question, because we're not entirely sure that all paraconodonts are actually phylogenetically like related. They might represent a more kind of polyphyletic assemblage of elements. So that's why we want to understand better how they're growing and compare them with like other conodonts and other vertebrate kind of tissues and structures in order to kind of test that hypothesis. But yeah, that's definitely a possibility.
Super, thanks very much, Will. So with that puzzle, we'll leave session 13A. I'd just like to thank all the speakers once again, and for all of you for some brilliant questions in the chat. So we've now got a 10-minute break, and then please come back for the penultimate session, session 14, which will be starting at 3 o'clock. So thanks very much. Bye for now.